Thank you for that excessively kind introduction. It really is a pleasure to be able to come here and share some of the work that I've been doing for a little over 10 years now. I know that a lot of you are actually interested in this area. I'm collaborating with many of you, and so it's nice to be able to come over here and give an update on what we've been doing. So my area of interest is in the realm of extracellular vesicles, and in general with a focus on diabetes and cardiovascular disease. That's what I'm going to talk about today. And I got interested in extracellular vesicles during my pharmacology training at Western. And it was really in the sort of from the concept of intracellular communication. And when you think about biological systems, they have really dozens and hundreds of sort of microscopic and submicroscopic entities in them. But particularly at that time in 2008, an area that was really relatively understudied was this area of extracellular vesicles. And so extracellular vesicle, a simple definition, is a particle that's released naturally from a cell that is surrounded by a lipid bilayer and can't replicate. And so that was sort of the consensus definition that the International Society of Extracellular Vesicles came up with just a couple years ago. And there are really three well-described classes of extracellular vesicles. There are other classes that are described, but really three well-described classes of extracellular vesicles. And we're going to explore these a little bit in depth, each of them. So there are exosomes, which are sort of the smallest class of extracellular vesicles. And they arise from endosomal sorting pathways and then are released into the extracellular space. And most of the data we have on these is that they're really designed for intercellular communication, designed to shuttle things from one cell to the next. You're probably also familiar with apoptotic bodies, which are sort of large particles which are released at the end stages of programmed cell death. And so these get released into the extracellular space, and they have all of the components that you can find in a cell. But an area that's, I would say, is still a little bit understudied, and I find most interesting is this intermediate class of vesicles called microparticles or microvesicles. And these arise from membrane blebbing under conditions of cell stress, and then are pinched off and released into the extracellular environment. And those ones are going to be probably the majority of the talk I'm going to share with you today. I will say a quick note about nomenclature. So those terms that I just used relate to the biological processes leading to the formation of those vesicles. However, in practice, most of us never actually observe the vesicle being formed. So we isolate the vesicles by centrifugation or adhesion techniques. And so we have a good idea of the size, but we don't actually know the biogenesis. And so there's been a push to use the term small extracellular vesicles or large extracellular vesicles in place of sort of these conventional terms. I'm going to focus on the classic terms for now, but just to note that I do acknowledge that I don't necessarily know that 100% of what I'm isolating is microparticles. There may be exosomes in that as well, although it's certainly enriched for microparticles. So I thought sometimes a picture is worth 1,000 words in terms of understanding these various vesicles. And so this is an image of late-stage apoptosis and release of apoptotic bodies from, I believe this is an endothelial cell. And so apoptotic bodies, as I say, they're 1 to 5 microns in size, very large. They have a significant amount of cytoplasm in them. They're shed exclusively from dying cells, and they have a semi-permeable membrane, and that is one feature that distinguishes them from other extracellular vesicle populations. So things will leak in and out of apoptotic bodies, whereas they won't from microparticles or exosomes. They contain all of the elements you can find in a cell, so RNA, DNA, protein, nuclear protein. Again, that's a unique feature to apoptotic bodies, is the presence of nuclear protein. And in general, the role in intercellular signaling is limited, and that's because they are very rapidly cleared in the body on the order of seconds to minutes. There are some exceptions, one of which is being studied here by Katie Rayner, which is when this efferocytosis process is impaired, like in atherosclerosis, then there may be a role for signaling for the apoptotic bodies. Exosomes are very, very different. They're formed through a multi-step process. So this is an electron micrograph of a cancer cell. I can't remember the line, but it's a very unique micrograph because it contains all three stages of exosome formation in the same picture. So if you look here at number 1, there's this membrane invagination of an initial endocytic vesicle. And then once that pinches off and accumulates inside the cell, you get gradual growth and fusion of these into something known as a multivesicular body, which you can see here with the star. And so the multivesicular body has a number of fates, so it can fuse with a lysosome where its contents are degraded, and so that's one way in which membrane protein is recycled within the cell. But alternatively, this multivesicular body can fuse with the cell membrane and release its contents into the extracellular space as 40 to 100 nanometer vesicles, and that's what's known as exosomes. So as I say, 40 to 100 nanometers, the definition varies a little bit. Some people will say 20 to 150 nanometers. They're actively formed and released from viable cells, contain protein mRNA, microRNA, but there's no evidence that they contain any nuclear proteins or nuclear material. Contain an impermeable membrane, and interestingly, the cargo is at least partially selective, so you get enrichment in proteins in exosomes where it seems as if they're being put into the exosomes by design. Likewise for RNA or in particular microRNA species. And the major focus in research right now is really in regenerative medicine. And so Daryl Davis is doing some of this. We've done some work in that. But there is really abundant evidence that if you take these exosomes or small extracellular vesicles from progenitor cells, and there's a variety of cell types you can use, mesenchymal stromal cells, endothelial progenitor cells. If you collect them and administer them to injured cells, they promote reparative processes. So they reduce oxidative stress, they promote angiogenesis, reduce inflammatory processes. So very potent. They seem to almost entirely recapitulate, in many cases, the protective effects of the cells of origin. And of course, that gets around many of the concerns with cell therapy as well. So it's been very appealing in regenerative medicine. In collaboration with Kevin Burns at the Kidney Research Center, we've actually done some work in this area. I'm not going to show a lot of it, but we have shown that if you take these small EVs from endothelial colony-forming cells, it's a form of an endothelial progenitor cell, and we treat them with an ischemic injury to the kidney, we can actually reverse the ischemic damage. So this is creatinine, so it's a measure of kidney function. If these kidneys are subjected to ischemia or reperfusion, you get an impairment in function and accumulation of creatinine in the bloodstream. Administration of exosomes at the time of reperfusion actually reduces creatinine, and after 24 hours has completely blocked the injury. And this is just some histology there. Some histology showing again a reduction in the tubular necrosis, cast formation in the tubules. So very beneficial and protective. Kevin's gone on to do a lot of work in this area, and this is sort of one example. But for the take-home message really is that these small EVs, these exosomes, appear to be protective and beneficial in a variety of acute and chronic diseases. I'm going to spend the most of my talk talking about these larger extracellular vesicles known as microparticles. And I don't want to say exosomes are boring, but I happen to find microparticles a little more interesting for a number of reasons. Most of which relates to the fact that they're formed directly from cell stress. And so this is sort of an overview of the membrane blebbing process. It's a conserved cell stress response. It happens in all cells in the body. And what you have here is you get nucleation, which is sort of a breakdown of the cytoskeleton immediately adjacent to the membrane. And you get cytosol flowing into this open area now. And it leads to this blebbing formation releasing from the cells. And this happens even with mild forms of cell stress. It doesn't actually have to be sort of a lethal or something that's going to lead to apoptosis. Even mild forms of cell stress cause this. And so once these blebs are formed, they can have one of two fates. They can sort of recover on their own through a process of recruitment of cytoskeletal proteins and retraction, or they can pinch off and be released into the extracellular space. And that's, at that point, you have what's known as a microparticle or a microvesicle. So this is sort of a summary of microparticle formation. Again, an endothelial cell here where you can see it's largely got the cell structure intact, but you can see formation of a few of these blebs. Another video image here where you're going to see a single microparticle pinching off of the surface of a cell. So as I said, they're also referred to as microvesicles. Sometimes in bacterial literature in particular, you'll see the term ectosomes. These tend to be around 100 to 1,000 nanometers in size, and they're formed really under conditions of mild to moderate cell stress. So you don't necessarily have to see apoptotic cell death. And this is just sort of one study that illustrated this fact. So this was Sapid in 2006 was generating microparticles off of endothelial cells treated with thrombin, and they saw a dose-dependent increase in microparticle release, even at doses where thrombin was not inducing apoptosis in those endothelial cells. And if they treated with a blocker of apoptosis, this DEV-D compound, they weren't able to block the microparticle release. So it's completely independent of apoptotic cell death, really just in this case, sort of in response to endothelial cell activation with thrombin. Another thing that's interesting about microparticles, so like exosomes, they contain microRNA, mRNA, protein. There's some debate, but most people think they don't contain nuclear material. But in contrast to exosomes, the content of these microparticles is generally reflective of the cell content. So this is just a summary, some proteomics we did from exosomes, microparticles, and cells. So EX exosomes, MP microparticles, CL cells, two separate lines. What you can see is actually when you do sort of unsupervised clustering, you get the exosomes very distinct, not much overlap at all, but you do get clustering of the microparticles with their cell of origin. And this is true for RNA and microRNA material as well. So you don't have this sort of enrichment process that the exosomes have. Microparticles are probably formed from all cell types, but when looking in vivo, we're really limited in terms of access. So we know that we can find microparticles in blood, urine, saliva, really any bodily fluid that comes in contact with cells. And so I think I've outlined this pretty clearly, but microparticles are emerging because of all of these properties as a biomarker of cell stress or injury. So they're released exclusively following cell stress. Their formation occurs even when it's not fatal cell stress, so at a time when you might be able to intervene and prevent cell loss. They retain many of the characteristics that they originate from, and one particular way that this can be exploited is looking at surface proteins. So you can look at, for example, an endothelial cell surface marker and note that they're coming from endothelial cells. And finally, they can be easily accessed in biological fluids. And so this is just a sort of a quick summary of some of the various microparticle species that we have looked at, and then the relevant biological processes in the biological fluid that they can be found in. So plasma mainly contains platelet microparticles, leukocyte microparticles, endothelial microparticles, and a small amount of erythrocyte microparticles. Urine will contain microparticles from any of the cells that come in contact with urine, so tubular epithelium, podocytes, that sort of thing. And then, various other situations, mesothelial cells we've looked at in the context of peritoneal stress, and then many investigators have looked at sort of cancer-derived microparticles as a indication of presence. So I'm going to talk, try and give you a couple of take-home messages about this. The first one is that circulating microparticles offer insights into vascular health. And this work really dates back to the start of my postdoctoral work with Rianne Tao, who Dr. Liu mentioned. So she was kind enough to let me work on an area that she wasn't at the time interested in. I sort of convinced her to get into it, sort of kicking and screaming over time. And we started looking at the levels of endothelial microparticles in hypertensive mice. So this was an animal model of hypertension where we treat with angiotensin II. We get a robust increase in blood pressure, about 40 to 50 millimeters of mercury. And in line with that, we saw evidence of increases in circulating endothelial microparticles, suggesting that there is an underlying endothelial stress associated with this hypertension. And of course, we know that from many other measures as well. We went on to look at what exactly was causing this in these mice. So we took cultured endothelial cells. So these are mouse aortic endothelial cells, and they were treated with angiotensin II. And so we showed that we could recapitulate this induction of microparticle release with treatment of angiotensin II. And then we showed that we could block this by treating with apocynin, which is an inhibitor of reactive oxygen species production, and by treating with fasodil, which is an inhibitor of Rho kinase, which is involved in cytoskeletal reorganization. So taken together, what we showed was that there's this oxidative stress leading to activation of Rho kinase, and Rhiannon had some evidence of that previously in her lab, but that in turn led to this cytoskeletal reorganization underneath the membrane, leading ultimately to the release of the microparticles. And so by treating with either an antioxidant or a Rho kinase inhibitor, we were able to prevent this. This is recapitulated in human disease. So this is a study from Wong a couple years after us, right around the same time of us, where they showed that uncontrolled hypertensive patients had increases in circulating endothelial microparticles. They also showed that if you treat with antihypertensives and control blood pressure, you get a reduction but not a normalization of endothelial microparticle levels. And they showed a very nice, robust correlation between blood pressure and endothelial microparticle levels. So it does seem to be a fairly sensitive measure of vascular stress in hypertension. The results are not, this observation isn't restricted to hypertension. It's been shown in a number of other diseases. We've looked in diabetes. So this is endothelial microparticles, circulating endothelial microparticles in wild-type mice and in OVE26, which is a model of type 1 diabetes. And we showed that there was about a 3 to 4-fold increase in circulating endothelial microparticles in this population. And again, at the cellular level, we could recapitulate this with exposure to high glucose. What's really interesting about this is that the levels of endothelial microparticles really do seem to correlate well with other measures of endothelial health. So I showed blood pressure, for example, in hypertensive individuals, but also there's robust correlations with aortic pulse wave velocity. So that's a measure of vascular stiffness. And there's a nice correlation, higher endothelial microparticles associated with higher levels of vascular stiffness. And there's an inverse correlation between microparticles and flow-mediated dilation of blood vessels. So higher endothelial microparticles in this case is associated with an impaired ability of blood vessels to relax. So it's nice to know that there's associative relationships between microparticles and vascular health. But of course, the first question that comes up, especially from somebody with a pharmacology background, is what are these things doing? Do they have a signaling role themselves? And so again, this was sort of one of the earliest questions we asked, and we looked at whether microparticles actually had a role in signaling in the vasculature. And in fact, they do. So if you expose endothelial cells, cultured endothelial cells, to endothelial microparticles, and the dose we used is similar to what the levels in our hypertensive mice, so it's a sort of a pathologically relevant level of endothelial microparticles. We get a very robust induction in oxidative stress with 3 measures here, so superoxide anion production and hydrogen peroxide production. So there's an induction in oxidative stress. We also get an inflammatory response with upregulation of cell adhesion molecules like VCAM and platelet endothelial cell adhesion molecule, and a tendency for macrophages in a flow system to adhere more strongly to our endothelial cells. And if we expose isolated blood vessels, in this case, these are mesenteric arteries from mice, if we expose them to microparticles in the water bath, we actually see an impairment in acetylcholine-mediated relaxation of our blood vessels. So a normal relaxation happens around 10 to the 7, 10 to the 6, somewhere in there we begin to see it. We actually, it's almost a full log, almost a 10-fold impairment in relaxation of our blood vessels when they're exposed to these microparticles. So what about practical applications for this? How do we take this knowledge and benefit patients? And so I wouldn't say we were the first to establish practical applications for this. That was probably Nikos Werner's group in 2011. What they did is they looked at individuals with coronary artery disease but no history of myocardial infarction. And they divided those patients at baseline between those that had high levels of microparticles or those that had low levels of microparticles. So that's above or below the median. And what they found was that those individuals who had high levels of microparticles at baseline had a much greater incidence of cardiovascular events, so myocardial infarction, stroke, or some sort of venous event. than those with the low levels of endothelial microparticles. And then actually these cohorts, if you go into the study, they were actually fairly well matched. And so this was almost like it almost behaved as an independent marker for risk in that cohort. And that's been validated by other groups. So this is Nicolas Amabile, Chantal Boulanger's group in Paris, looking at levels of microparticles in individuals with chronic kidney disease. In this case, they had a larger cohort, so they divided their microparticle levels into tertiles, so lowest, middle, and highest. And what they found was that the highest levels of endothelial microparticles, again, associated with an increase in both global and cardiovascular mortality over a 6-year follow-up. And that wasn't really seen with other populations, so they looked at platelet microparticles as well, and that wasn't really the case for platelet microparticles. So we've begun to look at this as well in a bit of a different setting in the context of diabetes and pregnancy. And the rationale is that women with diabetes and pregnancy have a much greater risk of adverse pregnancy outcomes, preterm birth, preeclampsia, many others. And it's believed that maternal vascular health is one of the major determinants of these adverse outcomes. And so we just asked the question, if we look at in surrogate marker of vascular health, these endothelial microparticles early in pregnancy, can we tell anything about the outcomes in these women down the road? And so for this, we used archived specimens from a large randomized controlled trial ran out of Toronto called the CONCEPT trial. So women with type 1 diabetes, about 150 patients. So the first thing we learned was that there was some relationship between endothelial microparticles and blood pressure. It wasn't very strong in the population, but there was a correlation that was significant with blood pressure for our endothelial microparticles. We also saw one for leukocyte microparticles, interestingly, and there has been some suggestion that leukocyte activation may play a role in hypertension, but mainly the focus has been on endothelial activation. We also saw a relationship between endothelial microparticles and glucose levels. So this is mean glucose, which was taken from a glucose monitor on the patients. And there was a correlation, albeit somewhat weak, between levels of endothelial microparticles and mean glucose. And there was also a correlation between measures of glucose control. So this MAGE parameter is something that they use from these glucose monitors as a measure of glucose control. So higher MAGE means worse glucose control. And there was, again, a positive correlation between the microparticles and glucose and poor glucose control. But most interestingly was when we looked at whether or not levels early in pregnancy have any bearing on outcomes. So we did, much like the Sinning paper, we looked at dividing our patients into low levels or high levels, so above or below the median of endothelial microparticles. And we found that those patients that had high endothelial microparticles, so above the median, had about a 2.5-fold increased risk of NICU admission, had about a 6-fold increased risk of neonatal resuscitation, and a 2-fold increased risk of cesarean delivery. And even if we adjust for known risk factors for these things like glucose, blood pressure, maternal age, in fact, those relationships only get stronger, not weaker. So in that case, you're actually at risk of a 5-fold increased risk of NICU admission. So there does seem to be some predictive value by looking at maternal vascular health early in pregnancy. I'm going to talk a little bit about a project that has a bit of a Heart Institute connection. We've been working with Ben Hibbert on this recently, looking at individuals with end-stage kidney disease. And so end-stage kidney disease is when you have less than 15% kidney function, and at that stage, a patient requires some sort of renal replacement therapy, ideally ideally a kidney transplant, but failing that, some form of dialysis and hemodialysis, where the blood is removed from the patient and cleaned in a dialyzer extracorporeally, is the most common form. It's about 20,000 Canadians each year who undergo hemodialysis. It's about 1% of our total healthcare expenditure. And hemodialysis does a good job of replacing kidney filtration, but it doesn't completely replace kidney function. And one of the main things that this manifests in is in cardiovascular disease. So individuals on kidney dialysis are at about a 30-fold increased risk of cardiovascular disease. And there's a number of reasons for this: hypertension, vascular calcification, but at the end of the day, it's all due to vascular stress, and so there isn't a normalization of vascular stress. And so with Marcel Ruzicka over at the Riverside Hospital, we looked at microparticles in individuals on hemodialysis. So we looked at platelet, leukocyte, erythrocyte, and endothelial, excuse me. And what we found was that individuals on hemodialysis had a higher level of circulating endothelial microparticles than those than healthy individuals. And in fact, we also saw the same thing with platelet microparticles, a significant increase in circulating platelet microparticles compared to healthy individuals. This was not a new result, actually. In fact, we were mostly just validating some work from Chantal Boulanger's group about a decade earlier, where they saw significant increases in endothelial microparticles, as well as platelet microparticles in their end-stage kidney disease population as well. But what was interesting is when we looked to disaggregate the data by sex. And so if you look at the endothelial microparticles, the elevation in men and women appears to be about the same, because circulating levels are very similar between men and women. But in women on hemodialysis, we saw about a 2.5-fold, almost 3-fold increase in circulating platelet microparticles relative to men, and both of those populations are higher than the general population. But there is this sort of, sex-specific effect where women seem to be more susceptible to this platelet microparticle formation, and many people think platelet microparticle formation is a surrogate for platelet activation. Why does this matter? Well, in fact, if you look at the epidemiology, I think most people are familiar with the fact that women have a survival advantage, and this is across almost all ages in the population, across pretty much the entire world. So these dark lines here are mortality of men versus women. So above the line, it means men have a higher mortality. But on hemodialysis, it turns out that women actually, their survival advantage is completely wiped out. And of course, that means that women are actually doing worse on hemodialysis than men. And so we think that possibly there's something going on with the subclinical platelet activation that may partially explain this, and so we're sort of working to explain that right now. As it happens, it's not just a change in the count, the numbers of platelet microparticles. They're also a change in bioactivity. So we compared the procoagulant activity of microparticles from men and women, the same level of microparticles from men and women, and in fact, the platelet microparticles coming from women are actually have a higher procoagulant activity as well. So there seems to be this change where there's more of them, and they're more prone to promoting thrombosis. And so the last take-home message, and I sort of just alluded to it there, is that microparticle contents and bioactivity are altered in disease, and this might be able to provide some insight into the changes from their cell of origin. And so it may not be just the counts that matters, but it may be what's inside these microparticles that's playing a role as well. So this is some work that we did a couple years ago and just published in Diabetologia, looking at the protein composition of microparticles formed under different conditions. So these are endothelial microparticles formed in culture under normal glucose conditions, which is NG, high glucose conditions, which is HG here, and then an osmotic control of high L-glucose here. And what we found was that there were significant differences in the protein composition of these microparticles. And if we looked at the proteins that were enriched in our high glucose population, one of the things that jumped out at us is actually most of the proteins, when you do gene ontology, end up being things that are involved in cell metabolism. And so it does actually look like we're able to identify a metabolic stress on these cells just by looking at the protein composition of these microparticles. We've also gone on to start to look at this in vivo. So we've isolated microparticles from healthy mice, hypertensive mice, and diabetic mice here. And again, we get a sort of a segregation of proteins to the various conditions, with some being exclusively found in diabetes, some being exclusively found in hypertension, and then those that are sort of found in multiple disease conditions. And what's nice about this is if you do, again, a clustering, an unsupervised clustering, one of the things we saw is that the three populations do segregate nicely. So our wild type populations all partition together, our hypertensive populations all partition together, and then our diabetic populations partition together as well. So it does seem that at least we have some signature that is disease specific. And so we're at the very early stages of looking at some of these proteins, but a couple of them sort of became interesting in terms of their role in vascular disease. So for example, in type 1 diabetes, we found that arginase was really only found in our 3 diabetic mice and not in the hypertensives or the wild types. We found that angiotensin converting enzyme was present in both the wild type and both the hypertensive and the diabetic, but not the wild type population. So we don't really have, we haven't really taken this very far at this stage, but it does suggest that we may be able to identify sort of protein signatures that are associated with the vascular changes in these diseases. So I'm going to stop there, but I'm just going to give a quick summary. You may not know who this is, but this is a picture of US President Andrew Hamilton. And the reason I pick Andrew Hamilton to show you is that he was born when his mother was on her way back from his father's funeral, and he was in an area so remote that they don't actually know what state he was born in. It could have been one of 3 states. And like Andrew Hamilton, microparticles are formed under stressful conditions. So they can be found in the plasma, and our evidence suggests that they seem to reflect vascular injury. I didn't show this today, but they also can be found in urine and reflect kidney injury. And assessing elevated levels of circulating endothelial microparticles may actually provide insight into vascular changes and be predictive of cardiovascular risk. In addition, microparticles do have a signaling role, and they generally communicate, at least in the vasculature, in a negative fashion, promoting endothelial cell injury. And again, in the context of urinary microparticles, they also promote tubular fibrosis. And as I've just shown, this process may be altered in disease. So microparticles are also able to promote sort of negative behaviors. So with that, I'm going to stop. I'm just going to acknowledge the people that did this work, primarily Fengsha Shao and Akram Abulbagai, who are current members, and Madison Turner, who graduated last year from my lab, and the many collaborators that I work with in the funding sources. Thanks.